And for those who joined recently, and I am Gitai Kim, Director of the UCLA Human Stem Cell and the Genome Engineering Center. And our center focusing on the collaborative independent research and also core service in iPSLD programming and then genome engineering. So this education program is supported by the California Institute for Regenerative Medicine in collaboration with the industrial partner, Thermo Fisher Scientific. And we have, like our center has very special relationship because we have an automation system and there also stem cell work and the cross collaboration with the Thermo Fisher Scientific as well as the Thermo Fisher automation team. And the participants who attended at least 6 of the 7 lectures will receive the certificate of the completion. So if you miss 2 lectures because you registered late, and then but still you can qualify by attending makeup session next year. So please continue to participate. And for the attendance tracking, and please log in into Zoom using your full name. And if not, please enter your full name and email address in the chat box. And if you have not registered yet, and then please follow the registration instruction in the chat box. You can search and the top, and then I left multiple linkers and then also some official materials and then those kind of things you can download. And the question is always welcome at any time. Just turn on your mic and then ask a question. And the benefit of this lecture and the education system is interaction between instructors and then also a number of people, including me, and then we will answer the question if you leave the question in the chat box. And also we will discuss lecture 6, mesodermal differentiation. And we are pleased to have special guest speaker, Dr. Glauco Dosa, and Director of the Global Application Development Bioscience at Grainer Bio 1, and who will present 3D biology, which is just bioprinting to the future. And then now I would like to hand it over to Omar and Tom, followed by Dr. Sosa. And then also during the Dr. Sosa's presentation, if you have any question, you can interrupt or just directly ask a question in the chat box. Thank you. Omar?
Excellent. Perfect. So let me share my screen real quick. Over here. Can you guys see my slides okay?
Yes, we
Excellent.
can.
Perfect. So let's get started. Again, thank you everybody for joining today. Over the past several weeks, we've been talking a lot about what stem cells are, where they originate from, how to work with these cells, potential applications, and kind of going through what we call the stem cell workflow, right? We talked about reprogramming. Tom and I talked to you guys about stem cell culture in general, what to look for in terms of morphology, quality of the cells. Tom did a great job as well last week of talking about editing in iPSCs and engineering, genome engineering them. And today we're going to focus mainly, or we're going to start to focus on the differentiation of these cells, right? The kind of one of the last steps of our stem cell workflow. And so before we get started talking about this, just generally speaking, I think there's a bunch of concepts that we have to introduce in terms of what differentiation is and what these cells are capable of in that context, right? So in order to help illustrate this, right, there's this picture or this diagram of almost like a tree branching, which I really I love this analogy because at the base of this, this tree or this the trunk of the tree is your pluripotent stem cells. Right? And if you remember from the very first lecture, we talked about how pluripotent stem cells have two main properties, right? Self-renewal and the capability of differentiating into all of the three germ layers that make up all of the cells in our body, right? But how does this happen, right? How does a group of cells or a population of cells know how to give rise to one cell type versus the other? And if we're using the analogy here of the tree, how does it know to go, and derive ectodermal lineages that can make up neurons, or mesoderm lineages that can make up, blood or cardiomyocytes, right? Which we're going to actually talk about today. So in this context, it's really important to know what factors govern the differentiation process in general, right? And this could be a whole bunch of different things that include paracrine and autocrine signaling. So signals that the cells are, releasing out to themselves as well as their neighbors in terms of signaling molecules and growth factors. Um, this also happens in the for the developing embryo, right? In vivo, it happens in the context of the maternal womb, right, or the uterus. So there's maternal cues and hormones that also govern this process. But even think about things just as simple as physical forces, right? Stretches, cell-cell contact, ECM composition, or extracellular matrix composition, right? All of these things are factors that basically govern how these cells react to their surroundings, right? And including oxygen tension, right? Just how much oxygen is available to these cells can help dictate whether they become one cell type versus another, right? And um, I love asking folks this. So please, in the chat, you just go ahead and maybe flood the chat with all of the signaling molecules or metabolic pathways that you know or are interested in. Because when I first started learning about cell biology in general, we started learning about seven major signaling pathways, right, that govern how exactly HIF, VEGF, WNTs, AKT, FGF, BMP, JAK-STAT signaling, MAP kinase signaling, right? These are just some of the pathways that dictate how the cells respond to these external factors, and they start to change their kind of their transcriptomic signatures in order to kind of respond to external stimuli, right? And again, when I started, there was seven really main signaling pathways, but nowadays I think that it's upwards of 14 plus. Some people can claim even more. And these pathways interact with each other in a very, very complex way in the cell, right? In a single cell, to the point where, at the end of the day, they end up establishing these metabolic pathways, right? Where the proteins that are expressed in that cell, particularly, end up governing its function, right? Certain proteins will carry out certain functions, so they are expressed as dictated by the signaling pathway that the cells are responding to. Then you could have a cell of a different function, technically speaking, right? And this is how the process of differentiation kind of occurs in a very kind of simplified way, if I will. Now, that said, right, today we're going to specifically be talking about mesoderm as a lineage specification. Now, mesoderm is, as the name suggests, comes from the Latin prefix meso, meaning middle, and it's actually derived from the middle of the epiblast, right? Now, we all know that we've mentioned this before, that the inner cell mass in a growing blastocyst is the population of cells that resemble or have that pluripotent property, right? The ability to give rise to all different cell types. Now, the inner cell mass will kind of, as the embryo starts to grow, will differentiate into two distinct populations, right? The epiblast and the hypoblast. And if we look at this post-implantation embryo, this is depicted slightly here, where you can see the epiblast is closest to the polar trifectoderm, and then the hypoblast is closer to the blastocele cavity, right? And those two distinct populations will also then give rise to distinct subtypes of cells, right? And this is how the process of differentiation works, right? So in the epiblast, as you see here in a gastrulating embryo, the epiblast starts to invaginate, right, and kind of turn in on itself a little bit, creating the neural tube, and in at the same time also dictating or creating the mesoderm, as you can see over here. So in the mesoderm, which is this layer in the middle, right, that forms, there is that is also then derived into multiple different subpopulations of cells that give rise to the lateral plate mesoderm, the intermediate mesoderm, and the paraxial mesoderm. Now, this is very involved in terms of developmental biology, right? And you're not expected to know this specifically. But what I do want to point out is you can see that from these different lineages, you can get different cell types that arise, right? So from the lateral plate mesoderm, you can derive cardiomyocytes, heart and blood vessels, blood, connective tissue, limbs. And this is the lateral plate mesoderm that's right here, right? That forms. But from the intermediate mesoderm, the kidney and the gonads form. And then the paraxial mesoderm is where you get skeletal muscle, vertebral joints, cartilage, and skin also forming, right? So again, depending on what cell type you want to be working with or want to, study, you have to make sure that you're closely mimicking this early embryo development of leading, your pluripotent stem cells down the right quote unquote branch, if you will, in order to get to your terminal endpoint, right? And specifically today, right, when we're talking about mesoderm, we're going to focus on one particular cell type that is of great interest for many folks, and that's, cardiovascular cell types or cardiac cells. Right? This usually is a differentiation that involves the activation of several different pathways, including Wnt, FGF, BMP, and sonic hedgehog amongst others. Right? Any questions so far? I should mention, as part of the mesoderm, I personally studied female reproductive tract development during my graduate work. And again, I wanted to mention this because it was Mother's Day, not too long ago. So happy Mother's Day to everybody as well. Okay, but focusing on cardiomyocytes specifically, right? Over the years, right, we've started to understand a little bit more about what these signaling pathways needed to be activated in order to derive our cardiac cells specifically. So in the early 2000s, we were using, feeder-dependent cultures. If you remember, those are the PSC cultures that are plated onto a mouse embryonic fibroblasts or feeders, and creating embryoid bodies, culturing these cells or these embryoid bodies in DMEM with 20% FBS, like we mentioned in our older systems, in order to derive these cells in about 20 days or so. But as we continued to understand a little bit more about the signaling pathways required in order to derive mesoderm and specifically cardiac cells, right, we started to take a more targeted approach where we'd use either small or growth factors needed along the way, like they do in the Gordon-Keller protocol, or small molecules in order to again achieve the same thing, right? Drive those signaling pathways specifically with the Pellicek protocol, where we're using small molecules to activate Wnt and inhibit Wnt signaling, for example, to drive this process. And then as these things became a little bit more, or we understood the signaling pathway a little bit more, you started to see suppliers like Thermo Fisher, for example, packaging all of these components that you need into ready-to-use kits as well. So these are also there's also commercially available kits that you can use in order to derive your particular cell type, in this case, cardiomyocytes. But if you think about it, again, the process for all of these approaches is typically the same, right? You're enacting a change on your pluripotent stem cell population that allows them to start activating these signaling pathways that are specifically going to derive your progenitors that you need, and then eventually your terminal cell type. And this is an example of using the kit that we have, for example, at Thermo Fisher, right? The cardiomyocyte differentiation kit. Our kit that we make is again not very different to all those other approaches in that regard, but we have two components in this kit, component A and component B, that are added at different times in order to again sequentially drive signaling in a very specific and targeted way. But at the end of a 2 week period, and I hope these videos work, you in a monolayer setting or in 2D culture, we get these beautiful wave-like beating cardiomyocytes that literally look like they're in a wave because they contract on one side, pulling on the other, creating this wave-like motion, right? And that is something that we can use in order to do downstream studies. But if you think about what cardiomyocytes are, right? They're muscle cells of your heart, right? And they should be contracting, right? That is the function of what a heart muscle cell should do, right? It should contract in order to pump blood. And that is exactly, again, what we're seeing here in terms of a functional assessment of what our terminal cell type needs to be doing, right? In this case as well, we can also track calcium transients, which are super important for this influx, which controls this contractility of these cells. And so we can follow these calcium influxes into the cytoplasm of these cells as they're contracting after our derivation or induction into this cardiac state. So this is sort of done in mostly in 2D or early on in 2D and 3D. But as many of you have probably heard, there's been a very concerted effort to try to also generate three-dimensional models that would be maybe a little bit more representative of our biology as humans, right? We're 3D beings, we're not flat beings. And so in order to properly recapitulate our biology, 3D systems or organoid models provide a really nice way to bridge this gap of how do we, really say that all of the signaling pathways that are important for cardiac cells based on a 2D model that may not necessarily represent us as humans, right? That said, I really also want to make sure that I get this point across that there is always a comparison of 2D versus 3D, right? And which one is better? And I think this is a much more complex question than folks realize now, where 2D systems are not necessarily bad, right? I want to make this very clear. They provide a little bit more control in order to study initial processes. But at this point in our research, I think the biological questions are becoming more complex that require a more complex model, right? And in this case, going to that 3D or organoid space is going to be beneficial to understanding a little bit more of the complexity at this point in our field, right? So just something to consider. And this is not just specific to cardiac models, right? There's a lot of different research areas that are moving to this 3D space within neurobiology, hepatic biology, nephrology, gastroenterology, respiratory diseases and biology, but even cancer biology is also, starting to realize the need for these more complex or 3D models. And Tom will talk more about this again for neurobiology and ectoderm lineages next lecture. But up until then as well, we do have a little bit of resources and support for you in terms of what are the different media systems and growth factors that are normally or commonly used with different organoid models and systems, right? Whether that be the brain, liver, intestine, kidneys, there's a further list that goes on. And again, today we're talking more specifically about cardiac organoids or going to be cardiac cells, but these resources exist. Now, before I jump into like the 3D cardiac story or kind of how we translated, our 2D system and are now attempting to generate 3D models as well. I want to give you an idea about how we normally develop 3D models in general, right? There's different approaches or methodologies that you can take in order to establish 3D cultures or organoid cultures. The one that most folks are familiar with are scaffold-based methods. This is basically when you take your cells and you get them to aggregate in a well, but also encapsulate them in some sort of matrix. Usually an ECM derivative or a basal membrane extract derivative, like Geltrex or Matrigel. And then you get these little drops, if you will, that you can culture in the right media with the right growth factors. And you can see at the bottom, these are just examples of different organoid models, like right, neural, lung, hepatic, where the organoid itself or the cells are encapsulated in this matrix, and it acts as a scaffold or supporting structure. Now, there's also scaffold-free methods. In fact, the first method of really culturing cells in what we would now call organoids it was in a scaffold-free method as a hanging drop. This is a way in which you can take your cells, put them in a very small volume of media and onto the cover of your culture dish, and that flip that over so the cells are hanging from the top, if you will. You can also now use with innovations in this space, use low attachment plates that don't necessarily support the attachment of the cells to the bottom of the plastic, but rather they would rather aggregate and form these 3D structures themselves. You could do that in larger plate formats or smaller plate formats. There's some also really cool ways in which you can coat cells with magnetic nanoparticles and via with magnets basically control where these cells land and so how they move. And there's also suspension-based methods that are in spinner flasks, for example, or bioreactors, where cells are aggregating, but they're also grown in some sort of constant agitation, where you keep the cells moving as they are, in 3D or in suspension in some sort of media. Now, for Tom, again, we'll talk a little bit more about this next week. I just wanted to kind of give you an idea and prep you up now. But in some of those models, there could be a matrix that's added in these suspension-based models where cells are growing on these agitation or in agitation or while they're being rotated, where you can add a little bit of extracellular matrix, again, like Geltrex or Matrigel, directly into the media, where you're not necessarily encapsulating or covering the entire organoid, but you're giving the media just a little bit of rigidity to help these organoids form a little bit better. And again, Tom will cover some of this again with some of our neural models next week. But for today, we're going to be talking about cardiomyocytes, right, and mesoderm. So what's the benefit of moving to a 3D organoid system with cardiomyocytes? There's several reasons, and at least when we were thinking about this at Thermo of why we'd want to do this, we came up with a bunch of different reasons that are listed here, right? The first two are most relevant to biology, and the second two are more relevant to the process, and we'll talk about these two in just a second. But if you think about it, right, if we want to accurately model the way in which we develop in vivo, our hearts, are not made out of a single cell type, right? There's multiple cell types that are required in order for an organ to function, right, properly. And in this case, if we're going to be studying the way in which our heart develops, it is logical to think, well, in order to accurately depict disease models or replicate disease properly in the lab, we have to have a model that that has all these other cell types that would normally be affected, right? That does make sense. The second aspect of 3D biology here, which is super, super cool, is there is more cell-to-cell contact in these 3D models that can, again, be a little bit more of a faithful replication to in vivo biology, right? We talked about all the factors that govern differentiation, right? Like cell-cell contact, signaling or signaling gradients, morphogen gradients, oxygen tension. These things all vary in a 3D context, right? It's not a flat 2D layer, it's a complex 3D model. So all of these different factors will play a role in how the cells react and how potentially we can treat disease in those states if we understand these processes, right? So that's basically to do with biology. The second two, at least for on Thermo's end, we were thinking, well, how can we leverage existing tools that we have in order to generate these 3D models, right? And we had a very easy, what kind of easy transition or easy way to generate 3D models in general, because now we had developed our stem scale media, which allows us to already grow pluripotent stem cells in 3 dimensions, right? So if we already have the pluripotent stem cells in aggregates, then it should be a little bit easier for us to move into 3D organoids as well from these 3D aggregates, right? And because we have a whole bunch of media systems, growth factors at our disposal, we can also use some of our GIBCO medias, basal medias, growth factors in order to facilitate this transition from 2D to 3D. And this is exactly what we did, honestly. We took our stem scale PSC spheroids, if you remember from our previous lectures, where we're growing, again, our pluripotent undifferentiated stem cells in suspension, right? In the as free floating spheroids, again, in agitation or on these rotating platforms. And when we wanted to start the differentiation, right, we would then use one of our differentiation protocols or kits in order to do so. Now, we do have the cardiomyocyte differentiation kit, and it does work for 3D differentiation of these PSC spheroids. But what we've seen on our end is that the small molecule approach seems to work a little bit better in this 3D context with these spheroids. So in about a 2-week period, we can take these pluripotent stem cell spheroids in 2D and generate these. Yeah, hope this works. Can you guys see the video moving or
No.
no?
Omar, you just bumped out of your slide deck.
Okay. I think I see my Teams crashed, so I apologize for that. I knew something was happening. Just give me 1 second, I'll get this back up. I apologize.
All good.
I wanted to do this really neat trick with the microphone and the cardiomyocyte, but
I'm just glad I'm not the only one with technical problems, but mine are solved.
Yeah, he is the Dr. Clark Sosa, and he will present about 3D biology. Yeah. I will introduce him later.
I am so sorry. I should ask for a new computer. That's what I should do. Okay, I got it. Hopefully this does not crash again. So let's take another look.
May I ask a question?
Please.
If we're moving from the spheroids in suspension, they're considered 2D?
So the spheroids are considered 3D in suspension still, but the difference there is that they're still undifferentiated, right? In the
Yeah.
stem scale media, they retain their pluripotent status because in the stem scale media, the composition of the media is made so that it ensures that the signaling pathways are maintain that pluripotent state, right? But
Okay.
once you remove them from that media, and then you start adding or replacing the media into to have growth factors that would start activating the right pathways, then is when you'd start to see that spheroid slightly transform and change into your desired organoid type.
Okay,
Does that make
thanks.
sense? Of course. Okay. I'm hoping it's better now. Can you guys see the slides okay?
Yeah,
And I'm hoping you can see this now, these beating cardiomyocytes, right?
yeah,
And I don't know if this will work, but if I have a microphone and I show this, I usually like to tap on the microphone just to, let's see, you let me know if this works.
yeah, it is working.
It is
Beautiful.
working? Okay,
Yeah,
perfect. So the first time I saw this, I was sincerely in awe because if you think about what this means, it's in about 2 weeks we can get these pluripotent stem cell aggregates to form maybe very preliminary, immature, rudimentary hearts, right? These pumping little structures that look like they're contracting just like, hearts would do. And the protocol that we used initially was to take these stem scale spheroids and in a basal and an RPMI media that's supplemented with B27, making sure that it's B27 without insulin. We can start to add these small molecules that will activate first Wnt signaling with shear for about, 24 to 48 hours. And then we remove that around day 2, and we actually add the Wnt inhibitor in order to stop that signaling specifically. And these cells will continue to kind of differentiate into a mesoderm-specific fate before around day 6, where we will remove all of the small molecules that we added, and we'll actually supplement with B27 with insulin in this case to drive a cardiac mesoderm-specific fate as well. Now, for those of you who don't or aren't familiar with what B27 is, it's basically a supplement that was designed out of a lab in California, Greg Brewer's lab. B standing for the brewer, 27 standing for the 27th attempt at making this supplement. It's basically a supplement that was originally that was added to neuronal cultures to help neurons survive. But since then has been used in a bunch of different other applications and has been helpful for multiple different cell types in this case. Now, a couple of things I want to call out again about protocol differentiation or differentiation protocols and optimization in general. You'll notice that over here I included a graph where there seems to be a lot of variance, right, or a lot of variability in terms of TNNT2 positive cells, which is a marker for cardiomyocytes specifically. So this graph is basically saying how many cardiomyocytes do we get at the end of a differentiation protocol. And the reason why I added this specifically, and the reason why you see this big variance is a point I wanted to illustrate about the need for optimizing protocols, specifically when it comes to those signaling pathways, right? In this case, these small molecules, the duration and the concentration at which you add them at is extremely important, right? But once you optimize your protocol, you can get really efficient induction. In this case, in this experiment, we got over 90% cells that were of the right marker that we wanted at the terminal end of our differentiation. And I will say, this little video over here that you see at the top is actually, it's not an AI generated video. I promise this is an actual spheroid that or excuse me, an organoid that we imaged in the lab. It just happened to be the poster child because it was a heart inside of a heart and it was beating. So this became our poster child for cardiomyocytes and rightfully so, I would say. Now, this protocol that we kind of adapted from 2D to 3D, we did in collaboration with a lab at Harvard University. But again, not only is the concentration of the growth factors that was an important consideration in this case, but there were other parameters that you needed to consider. In our case, starting from the stem scale spheroids, we needed to consider the starting size of that spheroid, right? Do we start with very small pluripotent stem cell spheroids with our induction, right, at day zero? Do we start with potentially medium-sized spheroids or maybe larger spheroids? And again, at what concentration? Does concentration matter here of those small molecules depending on the size of the spheroid, right? And what we observed is this beating phenotype represented by these flashing hearts and their relative size seemed to be a little bit better with larger spheroids that were exposed to this agonist for a concentrated but short window, right? Is a general kind of takeaway here. Again, understanding this process just a little bit more. Um, and at the end, right? Oops. Um, oh, that's not working anymore. These videos are just not doing it for me today. I apologize. There we are. And then at the end, right, you can also see that our beating cardiomyocytes also seem to have these cavities that are being created or fluid-filled sacs. Again, not necessarily representation of or 100% representation of chambers, but at least an attempt or something that is, physically resembles that, which is a good sign at this phase. Right? And to this end, right, if we actually look at what is the outcome, if we follow the differentiation all the way through, using these smaller spheroids to start or these larger spheroids, what do we get? And if we follow the 14-day differentiation throughout, you'll see that these smaller spheroids don't really fare well once we differentiate them at that size. They mostly die. But if we start with pluripotent stem cell or stem scale spheroids here that are larger, we do see a better induction and a survival of these cardiac organoids. In fact, this is also supported by gene expression, where we do see, I would say, orders of magnitude higher expression of the cardiac specific markers at the end of our differentiation protocol if we, again, only start with larger spheroids as opposed to smaller spheroids in it. So when you're doing when you're transitioning to 3D culture, think about, all these different factors that could potentially impact how your cells react to the growth factors or media that you're placing them in. And this is not, this isn't work that has stopped, right? At least not for us. We're continuing to continuing to optimize our differentiation protocols as everybody should. And this is just another example of that, right? Where we described the protocol here with the stem scale spheroids using RPMI media that's supplemented with B27 and the small molecules approach, right? But we also know that we have a cardiomyocyte maintenance media that was specifically designed for cardiomyocyte biology, right? Could we swap this out? Could we swap out medias in this case, maintain the small molecules, but at least replace the RPMI and B27 with cardiac media, intended for these cells a little bit more. And in preliminary work here, comparing these two, this change of the RPMI B27 or the cardiomyocyte maintenance media in this protocol, we do see a difference, right, in response where we do see beating phenotypes of these cells at lower concentrations of this Wnt activator if they're maintained in cardiomyocyte media as opposed to RPMI, right? So again, the functional biology of this is also dictated by how well you optimize your differentiation protocol. Now, following the protocol, the development of these organoids themselves, right? I mentioned in about 2 weeks is when we get to our more mature cardiomyocyte population. But if we look throughout this culture period, from day zero onwards, we see that the cardiomyocyte development is quite mimicked well. So we do see markers of cells that indicate the derivation of the intermediary states. If we're going down that branch, right, we're hitting all the right nodes on that branch. The mesoderm, cardiogenic mesoderm, cardiac progenitors, immature cardiomyocytes, and then mature cardiomyocytes. And even though we can, specify these cardiomyocytes in about 2 weeks, we can maintain them in culture for a longer period of time. So we've taken these cardiomyocyte, or as we know now, at least at Thermo more commonly call them cardioids, up to 6 weeks in culture, where we can look at the population of cardiomyocytes that are, marked with cardiac troponin and alpha actinin seem to be in the center of these organoids for the most part. But you'll also notice that, again, very much like 3D biology or in vivo biology, cardiomyocytes don't just exist in a vacuum of their own, right, in our body. So there's other supporting cell types that encompasses organs. And in this case, we can also identify that these organoids also develop fibroblast cells that are vimentin positive that seem to create a stromal layer or a supportive layer, but also endothelial cells that seem to organize themselves around cardiomyocytes, which what we think is in an effort to try to vascularize, right? This is how hearts develop. It's trying to develop a vascular system, but potentially doesn't have all the tools or other supporting cell types just as of yet. Right? And in this case, right, after taking those 3D organoids and culturing them for a prolonged period of time, there's a bunch of different things that we can do at the end of that, right? We can take these cardiac organoids and dissociate them back down into 2D, plate them on these multi-electrode assays and measure their electrical rhythmic impulses. Okay? If you want to be a little bit fancier too, you can take the entire intact organoid and carefully place it on these electrodes, measure its rhythmic beat rates. In this case, we were able to measure a stable beat rate of 0.3 beats per second, right? For this little guy, for example, that you see right here. And if you have access to some really cool instrumentation, you can also start to look at T waves and depolarization patterns, right? Literally looking at whether you can use these cells in order to screen or to act as a model for hearts in very much the same way, right? So this is basically my kind of section for the cardiac story specifically. I have just a little bit more to talk about immune cells, but any questions around the cardiac differentiation or cardiac models?
Omar, there was one question from Sachin regarding percentages of troponin T in those 3D cells. I actually don't have that in front of me, but I can
it's
have
a
a dig
great
for it.
question. Yeah, it's a great question. This is ongoing work actually that we're trying to work on right now. It's a little bit trickier, or a little bit trickier
Why don't
said
you
than
answer the
done, right?
question?
What
Answer
is
the
it? Sorry?
question.
Was there another question? Sorry?
No, answer that one.
Oh, yeah. I will. Don't worry. It is a little bit easier said than done because we're trying to basically look at the makeup of these organoids in general and what they do, and how they kind of behave. They're a little bit different from one to one. That being said, we do have an instance where we can dissociate these cells a little bit more readily now, so we can look at single cell contributions, which is work that's ongoing right now. In the earliest settings, right? Excuse me. In the earliest settings, right? Not at 6 weeks, but around the 2 week mark, we do routinely get really high induction of troponin T2 positive cells under our optimized conditions. So we do typically see above 90%. But after culturing them, for extended culture in 3 dimensions, we see a shift in that in those percentages, but we don't know exactly what those percentages are just yet. I hope that kind of answers the question. And if I don't, call me out on it.
Melissa has a question.
Is there any neural component to the cardioids, like a sympathetic neuron
That's a good
innervation?
question. Nothing that we have assayed for, to be
Okay.
honest, Melissa. So I don't want to say yes or no because in this case, if we haven't looked for it, I can't definitively say that it's not there. But more likely than not, the answer would be no because ectoderm and mesoderm lineages can be quite different. So you can introduce neural cells to these, models
Hmm.
afterwards. People do that when they do organ on a chip or human on a chip where they connect different organs together, maybe even assembloids. There's definitely that could be done per se, but just from a lineage specification standpoint, it's probably unlikely. Of
Thank you.
course. Any other questions? Sweet. Okay, let's keep going. How am I doing for time? Yeah, pretty good. So we talked a little bit about cardiomyocyte specifically, right? But this is not the only cell type that is derived from the mesoderm, right? Another set of cell types, or if you will, like a group is blood cells in general, right? Or hematopoietic lineages, as we call them. And so these are also derived from mesoderm lineages. Now, specifically, hematopoietic cells or lineages have been of a great interest, especially in the past several years, because they can be used in order to, or be used in a way that can use our immune system to fight certain diseases specifically around cancers. They've been used quite a bit. Sure, a lot of you have heard about T cell or CAR T therapies, CAR NK therapies, things like that are coming up, where we can take these blood cells, right, whether T cells or B cells, even modify them in a certain way and put them back into our bodies, or into the patient's bodies, I should say, in order to specifically target cancer cells, for example. And this can be done multiple different ways depending on the source of the cell. In a what we call an autologous approach, a patient's own cells can be taken, reprogrammed. You can derive a pluripotent stem cell population. You can edit that population, if you will, before you differentiate them into this immune cell type or hematopoietic stem cell type and then transplant them back into your patient. But there's also another approach that you see here on the right, where the donor is different than the patient, where the donor, this is called an allogenic process, where cells from a donor are collected, processed. In this case, we take, pluripotent stem cell banks, we expand those. And again, you can potentially edit those cells if you want to, before you can use them in these iPSC-directed therapy settings. And specifically, immune cells have been targeted because of their efficacy, right? With iPSC sources, natural killer cells or NK cells have been a really, really interesting target cell type. And this is because these are, part of the innate immune system. They're innately cytotoxic, meaning that they can kill other cell types if they deem appropriate. Right? They can target malignant cells or antigen in an antigen-independent manner. They don't necessarily require HLA matching, which is a lot of lingo work, meaning that it can be an easier process to transplant these cells into patients without getting too much side effects from things like cytokine release syndromes or your own body reacting up again. So they make for a great off-the-shelf sort of therapy, right? And in this case, we can again use um, um, our pluripotent stem cell populations to derive these natural killer cells that can be used in these clinical applications as well, right? In this case, you'll see that we use a version of our stem scale media that is under our cell therapy systems or CTS umbrella. This is basically just a version of the stem scale media that's manufactured for clinical applications, which you don't have to use for basic research. We have both versions, but in this case, we're growing, the PSC spheroids in the stem scale media in very much the same way. But you'll notice here that the differentiation protocol is quite different, right? So in this case, we're deriving not cardiac mesoderm, we're deriving hematopoietic lineages and then eventually iNK cells. So we're going to start off with activating, Wnt and FGF signaling, VEGF signaling, and then move more as we get our hematopoietic stem cell or progenitor cells. We then move to specifically go down a lymphoid lineage with different growth factors and cytokines, mainly IL-15, IL-7, IL-3, c-KIT, and FLT3, right? So in this case, you're specifically trying to make a natural killer cell in about 38 days. And this is exactly what happens, right? If we're looking at the expression or the cell surface expression markers on these cells as they're changing, right? You'll see that in around day 9, when we expect the hematopoietic stem cell population or progenitor population to rise, we start to see the cell surface markers that indicate that, right? CD34, CD90 are proteins that start to emerge and are markers of these hematopoietic progenitor or populations. But as you get, further into the protocol, you'll start to see markers of natural killer cells specifically start to pop up, like CD56 and CD16, which indicate a cytotoxically active or an iNK cell that can actually target other cell types and kill them. We've done some more work here, which I won't go into too much detail into, to characterize these natural killer cells that we can derive, in terms of their cell surface markers, what they're expressing, and whether those indicate to us that whether these cells can be functionally active, right? Remember, a lot of these different cell types are categorized by function too, right? What do natural killer cells do? In this case, they target other cell types, right? As the name suggests. So in order to see whether, not just the cell surface markers, coincide with what actually a cell needs in order to be cytoactive or cytotoxically active. In theory, or not just in theory, will this work if we put this iNK cell with another cancer cell type, for example? And this is exactly what we did. We took these human colorectal GFP labeled cancer cells, and using a completely different kit that we have called the OncoPro tumoroid culture kit, we can culture these colorectal GFP positive primary cancer cells in a 3D format, and then we can add our iNK cells to those tumor cells to see if the GFP signal diminishes. Right? Ideally, what we want to see is those natural killer cells targeting and killing these other GFP positive cells. And so this is what we see, right? At time 0 is when we add or we co-culture these two cell types together, right? So just remember, GFP is indicating the presence of these human primary colorectal cancer cells. And the signal, as soon as we add the natural killer cells, the GFP signal starts to drop dramatically. And it does so in a dose-dependent manner, meaning the more iNK cells that we add, the faster this GFP signal drops, right? And if we stain these tumoroids, right, we could see cells dying that they're expressing caspase 3/7, indicating apoptosis and an active killing and targeting of these cancer cells by are natural killer cells that we derive from an iPSC source. And so with that, I'll get my section to kind of an end and pass it over to Dr. Sosa. But just as an FYI, there is a ton of resources that are at your disposal. You know, culture handbooks, e-learning modules, even as you know, as things like probes to look at, you know, what markers your cells are expressing. Are they the right state or phenotype? And all of these, you know, we're definitely happy to share with you all afterwards with the slides. But with that, I'll say thank you for listening, and I'll maybe take any questions before I pass it over to Dr. Sosa. Let's see. Any questions in the chat?
We're good, Omar. All caught up.
All good.
Great job. Thanks.
Perfect. Excellent. So in that case, again, I'll pass it over to Keita, maybe to you to introduce Dr. Sosa.
Okay, so let